And we're just slowly, Oop. Hi everyone, we're just slowly assembling for the antiretroviral for prevention science series, and we'll be starting in a couple minutes when we have a little more of a quorum. So please hold. And while you're waiting, if there are any questions you have for Professor Gandhi, if you'd like to put them in the chat, we'll be able to cover everything at the end of her talk. You'll give it another couple minutes. We're still seeing people joining the call. For those who are joining, we're just going to wait maybe another minute as people are still joining the call, and then we'll get started for the Antiretroviral for Prevention Science series. I think we'll get started. I'm Ken Mayer from the Harvard CFAR and Fenway Health, and on behalf of my co-convener, Dr. Patrick Sullivan of Emory University CFAR, we're happy to welcome you to the Antiretrovirals for Prevention Science series. Today, we're really fortunate to have with us Dr. Monica Gandhi, who's going to talk about optimizing HIV treatment and prevention through adherence measurements and interventions. Dr. Gandhi is a professor of medicine and associate chief in the Division of HIV, Infectious Diseases, and Global Medicine at UCSF. She's also the director of the UCSF Center for AIDS Research and the medical director of Ward 86 at San Francisco General Hospital. She is the associate program director at the fellowship at UCSF, and she's well known to all of you on the call, I'm sure, for her innovative work in trying to think about how we can best measure and intervene to optimize antiretroviral adherence, both for prevention and for treatment. So, Monica, thank you for spending time with us today. And at the end of Professor Gandhi's talk, we'll be able to have time for Q&A. And please pop your questions in the chat in the meantime. So, Monica, please take it away. Thank you.
Okay, well, thank you so much, and happy to be here on World AIDS Day with you all. So, we're going to just actually mention the history of World AIDS Day, and then launch right into adherence metrics and improving HIV treatment and prevention outcomes through scalable adherence interventions, including the use of long-acting agents. I think it's a moment in time to talk about history just for 5 minutes because it was June 5th, 1981, when the first descriptions of HIV came out in the CDC MMWR journal right now that we're concerned about. And it was an important day. And essentially, I really do want to mention that since this is Jane Goodall passed away on October 1st, 2025, that the history of how HIV entered human populations is really likely through the bushmeat trade, which was the practice of hunting primates for food, likely started in West Africa with that first entrance of the SIV strain into human populations, and then really the setup for conditions of spread in West Africa were really social disruption and colonialism and the establishment of the sex trade. And I want to read this quote, which is, "I've spent my life speaking out on behalf of animals, and there's one issue that stands out uniquely as one of the worst threats to their survival, which is the illegal wildlife trade," from Dr. Jane Goodall, who passed away 2 months ago. So this is where we are with HIV in the current UNAIDS update. We have 40.8 million people living with HIV worldwide, 31.6 million people on antiretroviral therapy. Remember, this does reflect statistics of 2024, so cannot reflect what has happened over 2025 yet. 1.3 million new infections per year. And it's really important for this talk to say that that is an unchanged number for the last 6 years. For the last three updates, there's been no decrease in incidence. Also, 630,000 deaths. Which in 2024 from AIDS, which is also unchanged over the last 6 years, and 44.1 million deaths from the beginning of the epidemic. This is nowhere near over, and there's a 73% virologic suppression rate worldwide on antiretroviral therapy. So we're going to talk about how to help increase that. Even prior to this very difficult year of 2025, there was stalls in funding for the HIV response. For example, PEPFAR under the Biden administration was not renewed for 5 years. It was just given a 1-year renewal. And there really has been an uptick in stigma and anti-LGBTQ sentiment, and it really was already starting to get difficult. And then there was 2025. Here in the US, we have about a 67% virologic suppression rate as of the CDC statistics in February 2025. Really, as like worldwide HIV clusters with poverty, interpersonal violence, in the US here, incarceration, and we have that epidemic in the South of 52% of new infections being in the South and Southeast, really mapping onto a map of poverty here in the United States. We don't, I think it is important to remember on World AIDS Day how difficult things were leading up to the establishment of World AIDS Day. This is the AIDS quilt laid across the Washington Mall in 1987. And it was really in response to that year, 1987, is when we got our first antiretroviral. It wasn't great. It was AZT. It was pulled off a shelf at the National Cancer Institute. But every other medication that we've gotten since then is really a tale of rational drug design. It's been incredible. We first had the NRTIs, then the NNRTIs, then the protease inhibitors. 2008 saw the first integrase inhibitor. And then we had some really great protease inhibitor at that point. Darunavir came about in 2008. And then fast forward to dolutegravir in 2013. In 2018, we got bictegravir, doravirine, and a single pill combination with a protease inhibitor for the first time. And in 2021, we got long-acting antiretroviral therapies. So it has been this trajectory that has changed the course of HIV worldwide, and we never want to go back. And it really was combination therapy that began in 1996 that started the trend of plummeting morbidity and mortality from HIV. And we are now coming on the 30-year anniversary of combination therapy for triple therapy, really, for antiretroviral therapy. In terms of national and global strategies, everything really changed the response from programs started here in the United States. A little boy named Ryan White, who had acquired HIV from a transfusion, being born with hemophilia, died in 1990. And later that year, a bipartisan act was funded by Congress, one of the first disease-specific programs in this country, and really changed the face of what we could do here in this country with the Ryan White Care funding for low-income patients. It really provides wraparound care, case management, social work for those living with HIV has been really transformative. And there are threats to this at this moment. In 2002, the Global Fund started to fight AIDS, tuberculosis, and malaria after a G8 meeting earlier that year. And in 2003, G.W. Bush, president, started the President's Emergency Plan for AIDS Relief, has saved 26 million lives to date. In 2010, we had a national response, which is called the National HIV/AIDS Strategy, started by President Obama. And then in 2019, the first President Trump administration actually started the End the HIV Epidemic Initiative. It was continued under Biden, and was very surprised to see all these unprecedented cuts of HIV research and programming in 2025 when that first EHE, which had been so successful, was funded and started under the first Trump administration. In terms of the history of World AIDS Day, the World Health Organization was started in 1948 following World War II. And it was really 1950 that the first World Health Day started in response to all the poor health that had resulted from such a terrible conflict around the world. And that is actually commemorated on April 7th every year. In 1982, there was a World Tuberculosis Day that was started on March 24th every year. And then in 1988, Dr. Jonathan Mann, who founded the WHO's Global Program on AIDS, and later, which later became UNAIDS, actually started World AIDS Day, December 1st of every year. Started in 1988 in a very difficult time and has been commemorated every year since. Has been a really important commemoration as we see that AIDS is nowhere near over. Started at a very difficult time. We now have the tools to end the HIV epidemic. And in 1991 and 92 and 2007, there were other days declared. All of these days are really important to remember the diseases and to work on resilience and responses. This year, the Trump administration says that they will not commemorate World AIDS Day. We'll see what comes out of the White House today. But um, actually, the idea that the State Department said is that Awareness Day is not a strategy. But actually, there have been other awareness days commemorated in this current administration, including World Autism Awareness Day, National Manufacturing Day, and it is a strategy in the sense that it started really in the idea that silence equals death, and commemorating and stressing the importance of this day helps us work on treatment and prevention even more. And so we're going to go into that next and really talk about prevention and treatment and adherence. I don't think this is going to happen if we can all advocate, but they're really under these current funding cuts of the U.S. administration. UNAIDS has put out a model that by 2029 we could get back to 2000 levels of HIV incidence. And what this is really reflecting is that although treatment has been taken on by a lot of countries in the setting of these unprecedented funding cuts this year, prevention seems to be more of a luxury, if you will, or less of being taken on by countries. And so it's really the concern is HIV prevention and how do we fund PrEP and all these incredible monumental really updates in pre-exposure prophylaxis. So this is what we're concerned about. And this was the America Global First strategy that was released on September 2nd by the current administration. And I would encourage you to read it. Actually, UNAIDS gave a response to the America strategy for global health on the 18th of September, and actually it really gave a more thorough response to that strategy on World AIDS Day just last week. In response to this, in a report called Overcoming Disruption, Transforming the AIDS Response. And there was a lot of heartening aspects of that global health strategy that the US released, in the sense that as countries take on their own responses, the idea is to still have ongoing US funding as we make that transformation for self-ownership of those responses. The problem was it occurred too quickly in this administration, and hopefully we'll be able to give given a reprieve and have more time for that that transition to occur more smoothly after this. And so we'll see. There was some heartening aspects in that report. Okay, so let's talk about optimizing antiretroviral therapy and PrEP and adherence monitoring and adherence interventions. So, adherence, and this is actually reinforced by a study in the ACTG called the A5381, or the Hakim study that's coming out in Lancet HIV this week, is really its adherence that has been somewhat of the Achilles heel of doing really well on antiretroviral therapy and certainly on pre-exposure prophylaxis. And we're not the only chronic infection or chronic state that suffers from long-term issues with ongoing adherence. It's been labeled America's other drug problem, and only for 51% of those who are treated for cardiovascular disease, for example, are have daily adherence to their heart medications, and so. But I think we're the field more than any other field that have been working on strategies to overcome the long-term adherence issues, and those strategies include interventions. They include packaging pills in that are multi-component into single pills, and now long-acting antiretroviral therapy. So drugs don't work in patients who don't take them, and so we are going to talk about adherence next. Now, as we talked about before, 73% is the virologic suppression rate worldwide among those living with HIV. That may have gone down in 2025 with all these disruptions from the US funding cuts. Here in the US, this is data only up to 2023. We're at about a 67% virologic suppression rate. And there are multiple adherence barriers. Forgetting is one, but I think it's really at least in our urban centers here in the US, it's really a combination of substance use, housing insecurity, and mental health illness. And there is a big component overlaid on the entire planet of stigma and of taking pills every day, and not and the stigma, the ongoing stigma of prevention and treatment in HIV. And so, sort of surreptitious use or long acting or ways to overcome that stigma are important when we think about, for example, single pill combinations or our adherence interventions. This was the data that was presented by the CDC in 2025 in February, showing that our ongoing rate, sort of, this is long-term adherence, is about 67%, our virologic suppression rate. So lower, actually higher among females, but really and higher in the Northeast, but lower in the South, and there's disparities all over the country. It's important to use the word disparity in HIV medicine. In terms of pre-exposure prophylaxis, that is true in a way even more so of PrEP, because in oral PrEP, people feel well, they're taking this as a preventative medication. And this was a systematic review published in Lancet HIV that 41% of those on PrEP across multiple settings globally discontinued within the first 6 months. And even those who stayed on PrEP, there was a suboptimal adherence rate. So about 37.7% had suboptimal adherence. And discontinuation rates were higher in sub-Saharan Africa than other regions. And importantly, discontinuation rates were lower in studies with adherence interventions that were worked in, which is why it's so important to think about different adherence interventions for PrEP and treatment both. So, how do we think about adherence interventions? Well, one thing to start thinking about before we talk about interventions is really how to measure adherence, because self-report can be fraught with social desirability bias and also recall bias. And certainly, just asking patients is absolutely the cheapest way, but not always the most objective way. And so, really, what gets measured gets managed. And so when we think about how to measure adherence, there really are different measures now, and this is really coming up in the literature. But subjective measures are really asking the participant in a study, for example, and more even subjective is also pill counts, because you can dump your pills before coming into a visit, and patient diaries, and retrospectively asking. And more objective metrics of adherence are pharmacy refill data if you have a closed system, though it doesn't guarantee that you've consumed the pill. Electronic monitoring and also doesn't guarantee on a pill box, on a pill cap, that you took the pill. And then pharmacologic measures are really measuring drug levels in a biomatrix in the body. And that's by definition objective. You can't get the drug in the body unless you've consumed the medication. And so, in terms of those in terms of pharmacologic metrics, where did they come about? Where did this all sort of launch, this idea of drug level monitoring? This really came about from the pre-exposure prophylaxis trials, because as these were coming out, and remember, IPREX was the first one that showed a 44% reduction in HIV incidence with TDF FTC versus placebo. That was a surprisingly low reduction in HIV incidence compared to the macaque models. And it really was because even in that clinical trial, adherence wasn't perfect. In fact, if you actually modeled the reduction of HIV incidence based on tenofovir levels in plasma in the IPREX trial, it was 96%, which is equivalent to the findings that we saw with TDF-FTC and macaque. So it means that even in the clinical trials, adherence was a problem. And Partners PrEP did well, meaning really reduced rates of HIV incidence, but there was a major motivator for adherence in that study in the sense that one partner who was living without HIV had a partner who was living with HIV. So serodiscordance is a major motivator for taking the pill. And then there were several other studies, and then there was FemPrep and Voice. And we all remember 2011, these trials were closing down because of futility. Really, these were the study of TDF FTC in young sexually active African women, and they weren't working. TDF FTC was not working for preventing HIV. And it wasn't that it wasn't working. Of course, TDF FTC works perfectly well in women, just as we saw in Partners PrEP, which is performed in men and women both. But it really was adherence that led to those reduced, the fertility or the non-efficacy that was seen with FTC TDF in those studies. For example, in Voice and FemPrep, self-report was 91 to 95%. Even return pill counts was high, but plasma tenofovir detection, because plasma had been left over from the study, and you can measure tenofovir in there, was low. In fact, fewer than a third of women in the active arm were taking the TDF-FTC daily. So it really shows that that adherence, even in the clinical trials, was what's going to limit the efficacy of pre-exposure prophylaxis. And it was really those studies and that idea of measuring tenofovir in plasma that launched the objective adherence monitoring field, really launched the idea of measuring drug levels in different biomatrices. And those biomatrices can be plasma, which is very short, meaning tenofovir levels in plasma measure short-term adherence. That's the same with urine. And then because tenofovir, and this is just unique to NRTIs, or in this case, a nucleotide reverse transcriptase inhibitor, which is what tenofovir is, it's intracellularly phosphorylated. And so tenofovir diphosphate levels inside cells like dried blood spots or peripheral blood mononuclear cells really give you longer term metrics of adherence over weeks, say 4 to 6 weeks. And then hair levels, actually, of any drug measures really long exposure to the drug, and it matters how much hair you cut. So a centimeter is about 4 weeks of exposure, 2 centimeters is 8 weeks of exposure. So it really can go on and on depending on the length of the hair. And all of these drugs are really stable in hair. So we got involved in the adherence monitoring field actually 2 decades ago in our lab at UCSF called the UCSF Hair Analytical Laboratory. And the idea of doing hair levels or tenofovir diphosphate in dried blood spots, though that doesn't work for cabotegravir or something that isn't intracellularly phosphorylated, is that they sort of smooth over. It's an AUC of AUCs in the sense that plasma levels can go up and down every day depending on when you took your medication or different day-to-day variation. But it's really a hair level sort of gives a long-term metric or an average level of exposure. And we compare it to hemoglobin A1C, for example. In diabetes monitoring, a single glucose level gives you one snapshot in time, but a hemoglobin A1C gives a more average level of glucose control. So we've been looking at hair levels for a long time in our lab, and actually in different studies, but I'll just show you one illustrative study, really showed that hair levels of antiretrovirals are strong, independent predictors of treatment outcomes. This is in the Weiss study. But that's not surprising because HERA really measures your adherence and it really measures your long-term adherence. It also actually has a pharmacokinetic aspect as well. Um, and HERA levels do increase following adherence interventions. So the problem with hair levels, actually, though, and with tenofovir diphosphate in dried blood spots and plasma levels of tenofovir, is that they all require lab personnel that are trained in doing liquid chromatography tandem mass spectrometry, because that's the technique by which any of these metrics are analyzed. And so after looking at hair levels, and all the groups looking at different biomatrices, we really wanted to think about a very quick and point of care metric to assess tenofovir at the point of care. And it'd be really nice to have a point of care adherence assay because then you can trigger that feedback or adherence interventions or SMS two-way messaging or pillbox interventions or peer support or motivational interviewing, all of our adherence interventions that we think about right there at the point of care. Now, the problem is the point of care metric is not going to involve a liquid chromatography tandem mass spec machine. It really does involve making an antibody towards tenofovir because when we think of point of care monitoring for pregnancy tests, for example, that's an antibody that's developed against beta HCG, SARS-CoV-2 testing. It's an antibody developed against an antigen to SARS-CoV-2. So anything point of care is not really going to involve a machine, but you really need an antibody that you can put into a cartridge, and essentially it draws the medication away and gives you yes, no if you're taking tenofovir. And the reason, of course, we wanted to focus on tenofovir is it's the backbone of oral PrEP, but also the backbone of the majority of ART used worldwide. So we did develop, working with a company, a antibody against tenofovir, and it was really specific, meaning it doesn't pick up any endogenous nucleotides in the body. It took a long time to get that actually, but so it doesn't pick up adenosine, for example, which is what tenofovir resembles, and then did with other groups directly observed therapy studies and figured out the right cutoff for tenofovir to embed into this point of care urine test. And it's as simple, it's qualitative. It's yes, no if you took tenofovir in the last 4 days. But it is at this point very sensitive and specific when compared against quantification by ELISA, which is, a way to quantitatively look at antibody, with the antibody, look at tenofovir quantification, and with LCMS-MS. So we'll come back to the urine tenofovir test in a minute in terms of its utility as an adherence intervention. Certainly, adherence interventions have been developed over all these years, and it really depends on your setting. We have nurse-delivered home visits. We have two-way SMS text messaging. We do motivational interviewing with participants or patients. We do interactive computer-based adherence promotion or peer mentoring or peer-delivered interventions. There are adherence clubs and contingency management, giving people a little bit of incentive to take their medications. And all of these really depend on the context. They actually haven't been looked at as majorly cost effective, meaning they haven't been globally scaled because these are usually contextual adherence interventions. And there's been a lot of interest in providing drug level feedback of antiretroviral levels, looking at an ARV level, telling someone in a motivationally in a motivational way, great job. We see this in your system. How can we help you take this pill every day? Or having a hard time taking it. Let me see how I can help you. All of that. There's been a lot of interest in drug level monitoring of antiretroviral levels. But the problem is you don't want to take time. You don't want to do a plasma level that takes, say, 4 to 6 weeks and then tell someone 6 weeks later how they were taking their medication. You really, again, want that point of care metric. So after we developed this point of care urine tenofovir assay with the antibody test, we have been looking at it in the context of drug level feedback as an adherence intervention. And the first study that we did that was published this year was in Namibia with the CDC, and in this case, this was actually a pre and post intervention design, not randomized yet, and these were all participants who are on tenofovir, lamivudine, dolutegravir, TLD, the most commonly used therapy worldwide. They all were all failing TLD. They had all failed to suppress after at least one round of enhanced adherence counseling, which is the WHO recommended way to work on failure in the context of TLD. And then essentially, we took all of those participants who were failing TLD and had failed EAC for at least 3 months and gave them the urine test and actually did then adherence counseling around the urine test in that motivational manner that we described before. And by 6 months, actually, 92% were virologically suppressed. And by 9 months, that effect was sustained, even though we stopped the intervention at 6 months. And importantly, a counterfactual of if we had just used EAC alone was about a 44% virologic suppression rate in country. So it looked like the urine test, at least compared to what we thought would happen, really did improve virologic suppression rates. And there was a lot of acceptability of the test by providers and participants. They actually liked saying, oh, well, this makes me realize this is in my body. There's a point of taking this to see it in your body. And so it really was something that providers also thought was really easy to use and also made it more tangible to counsel around an actual objective metric. So that was a pre and post intervention design. And we now are doing a randomized control trial in South Africa after some delays with everything going on this year with the NIH. We have now really launched this study, which is going to compare in a randomized way EAC, the enhanced adherence counseling recommended by the WHO, versus counseling around the point of care urine test with tenofovir. And we are quite quickly now enrolling in this study. Unfortunately, there's been a lot of adherence challenges in the context of the last 10 months in South Africa. So we have also looked at the urine test in the context of PrEP. And in this case, because we had the hair measures and we didn't do a large scale trial yet. We're going to look at that as things sort out at the NIH. But this was a small pilot randomized trial looking at 50 people who were randomized in Kenya. They were all women. They were all in non-serodiscordant partnerships, meaning their partner wasn't known to be positive. And they were randomized to either just getting counseling for their PrEP, for their TDF-FTC versus counseling around the urine tenofovir test. And the only thing we could look at to see if this would work was a longer term metric of adherence. So, of course, we chose hair levels. And we did see that hair levels of tenofovir increased in the arm that got counseling around the urine test compared to sort of standard PrEP counseling. And so as things sort out with the NIH, hopefully we'll do a larger study in a randomized way with that as well. And we also, and this is, I'm just going to mention this because we're really talking about adherence interventions to increase treatment and prevention optimization in this talk. But I think that urine levels can also be a triage tool for resistance screening. We are, of course, concerned about HIV drug resistance worldwide. We're even more concerned about that in light of the funding cuts. And we were at about a 4 to 8% by the March 2024 WHO surveillance resistance report, 4 to 8% dolutegravir resistance among those who are viremic on TLD. And so if you don't take your TLD at all, you're going to fail with wild type virus. But if you take your TLD off and on, then you're more likely to be resistant. And so the idea was that the urine test could screen for who needs resistance testing. For example, if you have tenofovir in your urine and you're virologically failing, you're more worried about actually being resistant than if you don't have tenofovir in your urine at all. And so we're excited to look at the urine test as a predictor for resistance, and we'll be launching that study as well. And then I really do have to mention as an adherence intervention, long-acting ART, because long-acting ART is really this idea that we always wanted a long-acting medication to help our adherence. Look at the field of contraception. This is a field in which taking an oral pill every day, especially if you're an adolescent girl or a young woman, led to high rates of unwanted pregnancies. And when the long-acting contraceptive formulations came, implants or Depo-Provera every 3 months, unwanted pregnancy in those populations absolutely went down. This is true, for example, with antipsychotics and the treatment of psychiatric disorders. It can be hard to take that antipsychotic every day in schizophrenia, and so long-acting was developed to help with adherence. And also in substance use disorder treatment, long-acting injectable naltrexone or Sublocade to help treat substance use disorder. So we always wanted long-acting medications for those with adherence challenges, even though, as we all know, the long-acting combination that we have right now, the only one approved, which is a combination of an integrase inhibitor, cabotegravir, and an RTI, rilpivirine, was at least only studied when it came out, and the FDA in package insert still says for cabotegravir, rilpivirine, use in those with virologic suppression, those without adherence difficulties, because FLAIR, ATLAS, ATLAS 2M, which were the three registrational trials for cabotegravir and rilpivirine, studied those long-acting combination treatment in those without adherence challenges, in those who had been doing perfectly well on oral ART. So what's the evidence that we now have as long-acting ART as an adherence support tool? Well, we really have kind of two major bodies. One are two big studies in those with adherence challenges, but without viremia. And this was the one performed in the United States. This is coming out in the New England Journal this year by Adi Arana et al. And this is the 5359 or the Latitude study. And the design of this was not to look at those with ongoing viremia. The design was to take those with adherence challenges as evidenced by viremia in the past, get people suppressed on oral ART by giving conditional economic incentives, and then randomizing to long-acting ART versus staying on oral ART. And this study actually had to be ended early on February 11th, 2024, because, and was presented just 3 weeks later at CROI 2024, because there was a superiority to being randomized to long-acting ART, at least in terms of the outcome of virologic failure. So 7.2% virologic failure rate on those with long-acting Cabril, as opposed to 25% on those who went and stayed on oral ART. And the Impala study that was presented at IAS in Kigali, Rwanda, now a couple of months ago, showed the same thing. They all had adherence challenges, but had to be virologically suppressed before switching over to Cabril, and Cabril was actually non-inferior to oral ART. What about the evidence of those with viremia who aren't taking the oral ART at all or just cannot get suppressed on their oral ART? We don't actually have randomized controlled trial evidence yet of the utility of long-acting ART in those with viremia, not RCT evidence, but we do have a lot of demonstration projects, including very large one at Emory in the Pones Clinic, and also at Ward 86, where I serve as medical director. As soon as we got these long-acting ART regimens, we knew the criteria of the inclusion criteria of the trials to be virologically suppressed, but we started using them from 2021 onwards in those with viremia who couldn't take oral ART at all. And then when the real reason they couldn't take it is all those sort of urban concomitant challenges. And we've been publishing our data, and Pones Clinic has been doing that as well. And our last study was in March of this year, where we took the first 370 people who are on long-acting ART at Ward 86 up to October 2024, and a third had started with viremia of those 370 people. And those who started with viremia had high rates of housing instability, substance use, and had high rates of low CD4 cell counts. You're not surprised. Actually, our whole population did, but there were higher rates in those with viremia. And we found that at 48 weeks, there was really no significant difference in achieving virologic suppression in those who started with or without viremia. It was about 98%. So really high rates of achieving virologic suppression on long-acting ART if you started with viremia. And so our data, the data from Grady, the data from small studies at the University of Mississippi, the data from a study that we did in San Francisco on mobile homeless populations all contributed to the IAS USA guidelines and the DHHS guidelines. So are both two major US-based guidelines committees in 2024 permitting the use of long-acting antiretroviral therapy for those with viremia. Really, both sets of guidelines pointed out that it is these patient populations who cannot take oral ART for a variety of challenges that are most at risk of progression and death. And so it is really life-saving, and these permission to use long-acting ART in those with viremia was really gratifying to see in the guidelines, and I think should be cut out and provided to insurance companies as proof that we should be able to use them. This is not just here in the US. The WHO in Kigali did release their new HIV prevention guidelines, and they actually did put long-acting cabotegravir and rilpivirine on their guidelines for the first time worldwide. They didn't specifically say that this can be used in those with viremia. In fact, based on the CARES data, they said specifically among those who are virologically suppressed, but just like providers in this country have been using long-acting Cabril and those without virologic suppression, I'm sure as Cabril becomes available, which it does need to become available worldwide, it will be used in those with viremia. And certainly as a prevention strategy, it's very important to get to higher rates of virologic suppression worldwide. We all remember the day that when partner opposites attract and partner two, these huge cohort studies with lots of condomless sex between a partner who's positive, living with less than 200 copies per mil, a partner who's living without HIV. There were zero transmissions, undetectable equals untransmittable. One of our most powerful prevention strategies is U equals U. I do, of course, want to mention that long-acting will also benefit, PrEP will also benefit from long-acting strategies for pre-exposure prophylaxis. This was the latest look at how many people need PrEP worldwide. And again, I do want to stress that at this moment in 2025, we're most worried about PrEP and access worldwide because of the funding cuts. Looks like many countries, in fact, 31 countries have really taken on their HIV treatment provision for their populations, even since the funding cuts under this administration. But PrEP we're more worried about, at least in terms of oral PrEP, and we'll talk about long-acting in a minute. So out of 10 million people essentially who need PrEP worldwide, only 2.5 million people were accessing it even prior to the 2025 funding cuts. And here in the US, this is a CDC analysis from May 9th that 336,000 people are only getting PrEP, at least as of 2021, and we have about 2.2 million people who need PrEP in this country. So we need to get to higher rates of PrEP, and we're hoping long-acting PrEP will help us get there. We already had cabotegravir since 2021 as a long-acting strategy to use every 2 months for pre-exposure prophylaxis, with the HPTN 083 and 084 studies showing the utility in men, transgender women, and cisgender women, respectively. But we now have lenacapavir as of June 18th. I won't go over all this data. Certainly you have heard about it a lot, but the twice yearly lenacapavir versus daily FTAF or daily FTC TDF in Purpose 1 in cisgender women and in Purpose 2 in men and gender diverse individuals really showed high efficacy. There were zero HIV infections in Purpose 1. There was actually 2 new infections that got described later, but at least at when 50% had reached of women had reached that 52 weeks median, there were zero infections. And then there were later two new seroconversions in Purpose 2 1. And then in Purpose 2, there were two seroconversions to begin with lenacapavir, and then one more was described later. So lenacapavir was approved on June 18th for HIV prevention. It remains to be seen, I think, with the voluntary licensing that the drug company is providing, what's going to happen. Because again, we still need the global fund. We still need PEPFAR. We do need still those global private partnerships. And lenacapavir has been put immediately on the World Health Organization guidelines, as it has been put on the CDC guidelines as of September 18th. So I'll actually end there because I really want to get your discussions and questions for the last 15 minutes, especially for World AIDS Day. So thank you so much for having me to talk about adherence and measuring and monitoring.
Thank you so much for that talk, which is informative and inspiring and a little depressing in measure. And so we're going to open this up for questions. I'll ask one just to get us rolling, but I'll encourage people, if you want to either put your virtual hands up or if you want to pop a question in the chat to the group or just to me privately, and I'll read it. We have an opportunity to hear from Dr. Gandhi about I made a list of things that I want to talk about, so we might need an hour after this. There's so many provocative things. I guess I'd like to just start by saying you noted early on the importance of sort of the wraparound services, social work services to the success of the Ryan White Care Act. And I wonder what you think the vulnerabilities to those kinds of services are in the current policy environment, and any way to predict. I think part of our dynamic right now is trying to line up consequences with actions in advance. So what do you think about those particular social work services that you mentioned as being so pivotal? What are they vulnerable, and what would the impacts be if there are cuts? Do we know?
Yes, I mean, we actually absolutely know what the impact would be if the Ryan White Care Program was cut, because the interesting thing about the virologic suppression rates in this country is that virologic suppression rates are consistently higher among Ryan White funded clinics than they are in clinics that are not Ryan White Care funded, which is astounding because these are low income populations, but it really points to the importance of wraparound care, of case management, of social work, of even incentives to provide adherence support. I mean, really the whole point was to help people take their medications. That's what this talk is about. And Ryan White Care Program is a fundamental strategy to help with virologic suppression rates. So they're about 86% in Ryan White Care funded clinics versus 67% across the country. So Ryan White Care funding has been pivotal, profound, and it's been here since 1990, always re-ratified in a bipartisan manner. So it was pretty shocking this time when we saw the White House skinny budget on April 16th to see three types of cuts proposed. And this was coming off of the already cuts in foreign aid and the work stop order that was put on PEPFAR on January 24th. And then it got put back on January 28th, but only care and treatment and no prevention except for those with pregnant women, and then gutting of USAID. So the skinny budget disturbingly does propose some cuts in the Ryan White Care Program in parts A and B, which are the main parts that fund our low-income clinics. Now, as you know, we're on a continuing resolution until January 30th, and the NIH budget cut, at least, which is proposed to be 40% by the White House, didn't seem to have favor at all by the Senate Appropriations Committee that met on July 31st, or the House Appropriations Committee. Doesn't mean it's the whole Senate or House, but both Appropriations Committee were like nothing doing with cutting the NIH budget by 40%. So we're hopeful that with negotiation that the Ryan White Care programs won't be cut and these can be continued. I have to say I was just so surprised with the traditional bipartisan support of HIV. But as you know, the White House has been really setting the stage for a lot what's going on, and canceling World AIDS Day or not commemorating it was quite a shock last week, because and so it does seem that things get proposed that are terrible and then they're somewhat walked back. I do think that even the global funding is going to be better than we think if you look at that global first, America First global health agenda. So I'm hoping that Ryan White stays intact. It's so important.
Thank you. I'll give a moment if anybody wants to raise hands or pop questions in the chat. Ken.
Monica, that was a wonderful talk. Would you want to expand a little more about the use case for the urine test and the different scenarios that you think are most feasible coming online soon? And talk a little bit about cost effectiveness, what you think the cost of the test will be, and if you've done any kind of cost effectiveness analyses.
Yeah, so there has been two cost effectiveness analyses, including in the paper that's in the International Journal of Infectious Disease, which is we looked at the cost effectiveness of it. So it'll probably be, we don't know what the company is going to set because we had to work with the company, but they are telling us around $2 to $5 a test. And at that price, it would still be cost effective because it is, really, it seems to work better at least than other adherence interventions. There's something when we've done a lot of acceptability, feasibility testing of the urine test, and patients seem to really like it. They participants mainly, they don't feel judged because you really have to do the counseling in a very motivational way, very supportive way. But that's always been baked into our studies. And actually, a lot of people said qualitatively that it proves it's in their system. Like it gives them comfort that it's in their system, especially with PrEP, where you don't have a viral load to monitor. So so we think it'll be cost effective. We're still launching some more studies, especially the resistance screening studies, and everything got delayed with the NIH cuts this year before they'll market it. So we're hoping for marketing in 5 years, say, but we still need some more studies to do.
Great. I'll ask one more question that is sort of a bigger picture. I think many of us are trying to figure out the most productive ways to go about proposing science that will have impact. And you really, got to this beautifully in the talk. I just wonder, do you think there's any gain in thinking more broadly about the adherence issues for other chronic diseases? And either as a grant writing strategy or as a policy strategy, do we need to be thinking about, obviously everything's in the specific, and HIV is different than other, health conditions. But is there an opportunity to sort of make a broader case about some of these things like adherence, like home accessibility of devices or information that would have broader policy appeal? And are there specific things about HIV that need to be carved out? Or could this be an approach to try and broaden, like, one, the important impact of the work that you've led and described, but also just from a funding perspective, to look at classes of tests that might then be applied to different kinds of health conditions?
Yeah, I mean, thank you for asking that because I just think HIV really leads the way. I mean, in so much of adherence, when that's why I really wanted to point out that the WHO has said that non-adherence is really one of the main causes of people not doing well in terms of health. So chronic diseases, cardiovascular disease, we all know that in congestive heart failure, you have to take 4 different classes of medications, and there is absolutely not single pill combinations. They're trying, but then they've studied like maybe putting it in a big capsule that you have to swallow, but otherwise you're taking multiple pills a day. That's way back to 1996 for us. And we got to single pill combinations. The first one was in 2006, which is efavirenz, TDF, and FTC. And then ever since then, it's surprising not to have someone on a single pill combination. So we've always kind of led the way. There's a recent Nature paper to say how much HIV has informed other fields. I know we all believe that as HIV researchers, but it is the documentation of that paper. Steve Deeks is the last author and Lawrence Corey is the first. It's really important to read how much we've informed the field. So when I think about future studies, cardiovascular disease would really benefit from single pill combinations. They would really benefit from long acting, and they would really benefit from adherence interventions using point of care metrics or other strategies to talk to people about adherence. When we think about chronic disease, it's not because of processed foods. It really isn't. It's because of, it's because of just difficulty taking the handfuls of pills. So we've always informed the field. We're always going to inform other fields, and we're going to keep on going with that. And in terms of funding, I do think, and I really want to stress this, and I'll answer then um, the other questions in the chat because they're really good. But I really want to say this. When you look at the August 15th strategy from the NIH on what are the priorities of the NIH, so just put in August 15th, NIH Director 2025 strategies. There are 12 strategies listed that the NIH wants to fund. And we can ignore nutrition and autism, that's not our field, but there are others that are really important. And one is AI in there. We have to bring AI into our work. One is non-animal models. Okay, we have to think about organoid models. And if any group's doing that is in the cure world, it is actually HIV. And then importantly, there's only one pathogen mentioned in that entire long document, and it's HIV. And that pathogen is number 12 of the 12 priorities. And it says care, treatment, prevention, and it says cure. It actually says cure. So I do think HIV is going to be prioritized by the NIH. It may not look like how we want it to look like. We can't use the words that we think are so tremendously important in our field, but it is going to be prioritized. And I think that gives me a lot of hope, and I tell early stage investigators that all the time. And then, do you want me to answer? Because there's some very good questions here. I would say
Yeah.
to the long-acting treatment from Raj, the long-acting treatments for substance use with long-acting ART. We just did an analysis in our clinic of those 370 people on long-acting ART. Actually, there was a lot of psychosis in that patient population because mental health was such a big, important part of our problems with taking oral pills, and we weren't using the long-acting antipsychotics enough. And so we started just last month looking at our lists, and every person who's on long-acting ART, we've evaluated if they need long-acting antipsychotics. Those are every 28 days. So we switched them to a 28-day Cabril. If they were on 8 weeks, we put them back on 4 weeks, and we started giving them Abilify or long-acting risperidone every 28 days. So I think that's a really good, important combination to do. And we're going to do that with substance use as well with Sublocade next. That's our initiative for January. And then I do think that Rose, with more focus on wraparound services, can you speak with specific services? Really, case management has been so important to just bring people to appointments and to remind people of the calls, especially with long acting, because it's only once a month or once every 2 months. So our wraparound services are increasingly really focused on the case management, helping with adherence. It's why Ryan White Care Clinics do so much better with adherence. So I think calls and bringing people to appointments. And then these other ones, Sean asks about the funding environment, these other adherence interventions. These are all good, actually. It's just that they haven't been widely scalable because they may not be always as cost effective. A nurse visit is certainly more expensive than, say bringing someone in or using a urine test. So, but it doesn't mean they're not important. SMS is pretty cheap, and that kind of mobile health is important, and that back and forth SMS messaging. Peer support, we all know, is important. Motivational interviewing. They're just more difficult to get funded. And I will tell you this, that I think combining those interventions with a biomarker of adherence, I've seen has been easier to get funded. When you put in biomarkers, that kind of drug level measurement is a little bit easier than just providing a social behavioral intervention.
Monica, we want to thank you so much for such a tour de force. Really covered so many important topics so well. And thank you for making World AIDS Day more meaningful to all of us who are committed to this work. So really appreciate it.
It's my honor to work in the AIDS community. Thank you very much.
Thank you. And everyone,
Thank you.
Alberto put the names of our upcoming talks in the chat, and we welcome your participation. So see you again soon. Thanks, everybody. Take care.